This video will discuss research by Ken-Rong Wu and his co-authors titled Analysis of Therapeutic Targets for SARS-CoV-2 and Discovery of Potential Drugs by Computational Methods. This video is presented by Caroline Culpepper, Sydney Taylor, and Jara Whitehead. Coronaviruses, such as severe acute respiratory syndrome coronavirus, SARS-CoV, and the Middle East respiratory syndrome coronavirus, MERS-CoV, have caused severe outbreaks of pneumonia since the start of the 21st century. Coronaviruses are a family of zoonotic viruses that are carried predominantly by animals and can spread to people. To this date, there is no therapeutic drug that has been approved to treat humans infected with coronavirus. Recently, COVID-19, a relative of SARS-CoV and MERS-CoV, was discovered in Wuhan, China, and has quickly become a global pandemic. This graph demonstrates the severity of COVID-19 in the United States specifically. According to the Center for Disease Control and Prevention, the number of coronavirus cases in the United States has increased exponentially since late March, with a total of 395,000 reported cases of COVID-19 as of April 7, 2020. According to this research paper by Ken-Rong Wu and others, the fastest way to treat COVID-19 is to find molecules from marketed drugs that can inhibit the virus. For instance, a potential anti-coronavirus therapy could involve blocking the virus binding to human cell receptors, preventing the synthesis of viral RNA, or inhibiting the virus's self-assembly process in the host body. Thus, the goal of this paper is to identify therapeutic targets involved in these processes in COVID-19 and to predict potential drugs that can inhibit these targets.
So, the next question is, how did researchers accomplish this goal? In this study, a bioinformatic method of blast sequencing was applied to reveal homologies in viral genomes with SARS-CoV-2. Among all human coronaviruses, SARS-CoV-2 exhibited the greatest alignment with SARS-CoV. This genome alignment and prior research focusing on the structure and function of SARS-CoV proteins inspired homology modeling for the SARS-CoV-2 virus. Protein structures were predicted for prominent structural and functional proteins, such as viral papain-like protease, 3-chymotrypsin-like protease, RNA-dependent RNA polymerase, and spike, just to name a few. In order to identify possible drug candidates, structure-based virtual ligand screening was used, so that 21 targets were screened against compound libraries such as the zinc drug database, a natural product database, and an antiviral compound library. The resulting compounds were continuously docked to 21 constructed targets of COVID-19 that were represented in potential energy maps created by the Internal Coordinate Mechanisms, or ICM software. The potential drugs were ranked according to the ICM scoring system, using a potential of mean force score, or MF score, that provides an independent score of the strength of the ligand-receptor interaction. Strong ligand-receptor interactions received MF scores of less than -100 and were selected as potential drug candidates based on their high binding affinity with the target protein. Compounds that bound weakly to target proteins, or compounds that were visually bound outside the active site of the target protein, were eliminated as potential drug candidates. The first category of targets are virulence factors. Virulence factors are produced by viruses for two main reasons: immunosuppression, which interferes with the host's innate immunity, and immunoevasion, which allows the virus to go undetected by the host's immune system. One of the COVID-19 virulence factors is ORF7A. This protein works as an antagonist to BST2 or tetherin in humans, which typically functions to inhibit the release of newly assembled viral particles from host cells by tethering these budding viral particles to the host cell membrane. The inhibitory effect of ORF7A on BST2 or tetherin allows the virus to continue releasing new virions and thus infecting new host cells within the body. NSPs are non-structural proteins of the coronavirus. In host cells, they are involved in RNA transcription, translation, protein synthesis, and virus replication. The NSP1 virulence factor has been linked to two main functions in host cells. One of these is interacting with the host 40S ribosomal subunit, which induces host mRNA degradation. Another is inhibiting type 1 interferon production, which would typically enhance antigen presentation and production of immune response mediators like cytokines and chemokines. NSP3C, or non-structural protein 3C, is another coronavirus virulence factor, which binds with the host's ADP ribose and helps the coronavirus resist host innate immunity. The drug lopinavir was identified to have high binding affinity to the NSP3C domain with an MF score of negative 189.5, which according to the scoring method indicates this drug to have strong interactions with the protein active site. Lopinavir is a marketed antiviral drug used to treat HIV infection. The drug targets HIV-1 protease, therefore inhibiting the virus from properly assembling and cleaving polyproteins. A separate in vitro study showed inhibition of SARS-CoV replication at a 4 microgram per milliliter dosage following 48 hours of incubation. At the cellular level, these results give indication of the possibility of repurposing lopinavir to target SARS-CoV virulence factors.
The next type of targets are those that prevent virus RNA synthesis and replication. One of these targets is NSP12, which is an RNA-dependent RNA polymerase, RDRP. NSP12 is a conserved protein in coronavirus and an essential enzyme of COVID-19's replication and transcription complex. The RNA polymerase domain of NSP12 is located at the C-terminus of every polypeptide chain and has a conserved serine aspartic acid motif. Furthermore, NSP8 can synthesize RNA primers for the NSP12 RNA synthesis de novo. It can also complex with NSP7 to increase the binding of NSP12 to RNA. The molecular docking results show that a potential inhibitor of NSP12's RNA polymerase function is remdesivir, a nucleoside analog and an antiviral drug. Remdesivir binds to NSP12's RNA polymerase domain with high affinity, resulting in an MF score of negative 112.8, which represents a strong ligand-receptor interaction. Specifically, remdesivir binds to the RNA binding channel of COVID-19's NSP12. This channel is usually reserved for the acceptor template nucleotide. However, remdesivir binds to this pocket by forming hydrogen bonds with asparagine 497, arginine 569, and aspartic acid 684. Additional hydrophobic interactions with leucine 576, alanine 685, and tyrosine 687 may also aid in the high binding affinity of remdesivir to this active site. Thus, by binding to the RNA binding channel of NSP12, remdesivir inhibits transcription and the synthesis of viral nucleic acid. Not only does remdesivir bind to NSP12, but it can also bind to the type 2 transmembrane serine protease, TMPRSS2. The type 2 transmembrane serine protease cleaves a COVID-19 protein called spike that is essential for viral entrance into the host cell. In order to prevent viral invasion, Remdesivir binds to the type 2 transmembrane serine protease with an MF score of -109.4. Remdesivir binds to a positively charged allosteric pocket on the type 2 transmembrane serine protease, which is very far away from the active site. Asparagine 84 and arginine 405 form hydrogen bonds with the phosphate groups of remdesivir. There are also hydrophobic interactions between the pyrrolotrizine ring of remdesivir and tyrosine 131 and 401 of the serine protease, which further stabilizes the interaction between the two molecules. As of February 18, 2020, remdesivir is undergoing double-blind Phase 3 clinical testing in China.
Structural proteins are an obvious target in drug design because successfully attacking the structural proteins of a virus will efficiently and specifically slow viral growth. Spike is the main structural protein of coronavirus, and its function is to allow the virus to enter host cells. The spike protein of SARS-CoV-2 binds to the host ACE2 receptor that is present on lung cells, mediating virus invasion and determining the infection specificity of the virus to certain host tissues. Spike is cleaved into S1 and S2 by the host protease. These subunits have different functions. S1 does the actual binding to ACE2, and S2 mediates membrane fusion between the virus and the host cell. This means that for spike, there are two mechanisms that can be targeted. By targeting the S1 subunit, you'd be looking for a compound that binds directly to spike's receptor binding domain. This compound would hopefully be a competitive inhibitor of ACE2 binding. If a compound doesn't bind to spike's receptor binding domain, it can still affect the protein by binding in another area. Compounds that bind in other areas can still affect S2 function because the fusion of the viral membrane with the host membrane requires a significant conformational change of spike after ACE2 has bound. This opens up the possibility of finding a variety of compounds that can affect spike protein function. The binding of spike to ACE2 and the membrane fusion between host and virus play an integral role in the process of infecting host cells, so targeting its structural integrity and cleavage is an extremely valuable therapeutic strategy. Virtual screening results of small compounds against spike protein showed a diverse group of drugs with high binding affinity to spike. Antihypertensive drugs, antifungal drugs, antibacterial drugs, anticoagulant drugs, and some natural flavonoids showed high binding affinity. It isn't known yet if these compounds would actually affect spike function, but it is possible that they could target S2 subunit function. Virtual screening results of small compounds against ACE2 also found several possible drugs that could bind to ACE2, but none of them bind to the area of interaction between ACE2 and spike. These compounds may be ACE2 enzymatic activity inhibitors rather than inhibitors of ACE2 mechanisms in viral infection. That means these compounds may actually be damaging in treatment of SARS-CoV-2, as ACE2 enzymatic activity helps protect lungs from injury. While it's possible that drugs that bind in areas other than spike's receptor binding domain can still affect its structural integrity or its ability to bind to ACE2, a compound that binds directly to the receptor binding domain with high affinity would be valuable in that it would ensure that spike cannot bind to its receptor. This would be a compound affecting S1 subunit function. The only compound found that would bind to the receptor binding domain was hesperidin, a flavonoid found in unripe citrus fruits. It was predicted to lie in the middle of the receptor binding domain of spike. The sugar part of the compound was inserted into a shallow pit where hydrophobic amino acid residues form a pocket to contain the compound. When the ACE2 receptor binding domain complex and the hesperidin receptor binding domain complex are superimposed, you can see the overlap of hesperidin and ACE2, meaning the ACE2 receptor binding domain interaction may be disturbed by hesperidin. This is significant because hesperidin may be able to directly block the binding of ACE2, acting as a competitive inhibitor. Other important structural proteins include E-channel proteins and N proteins. The receptor binding domains of coronavirus E and N protein are needed for N proteins in host cells to bind coronavirus DNA. Detailed results of this part of the study weren't provided, but it was found that several types of drugs screened from the zinc database and the in-house natural product database had relatively good binding affinity to these targets. When screening and identifying targets of antiviral drugs, it was found that arbidol may interact with E-channel protein. Arbidol is a broad-spectrum antiviral used to treat upper respiratory tract infections caused by the flu. Studies have proven arbidol to be effective against SARS-CoV and MERS-CoV. Arbidol hydrochloride can prevent virus replication by inhibiting cell-host membrane fusion. It was found that arbidol can inhibit coronavirus up to 60 times at a concentration of 10 to 30 micromolar relative to a control group. The E-channel protein of SARS-CoV-2 is a possible target of arbidol, but further studies would be needed to prove whether or not it actually inhibits the protein's activity. Previous outbreaks of SARS-CoV and MERS-CoV launched research efforts to find drugs for the treatment of zoonotic coronavirus, but the current options for treatment are limited. SARS-CoV-2 and future coronaviruses will continue to jeopardize global public health, so finding a broad-spectrum inhibitor will continue to be a research task. Because there is an urgent need for treatment options for the current global COVID-19 pandemic, existing treatments for other diseases may be the best option. In this study, 20 homology structures of SARS-CoV-2 proteins and one human protein, ACE2, were set up for high-throughput ligand screening to find compounds that bind to the proteins with high affinity. Existing antiviral drugs were docked to these targets as well. Whether or not the small compounds found in the antiviral drugs actually work on these targets will need to be confirmed with in vivo and in vitro studies. The goal of subsequent research for this group is to solve all 24 SARS-CoV-2 proteins and their drug complexes, perform in vivo and in vitro studies, and prepare for clinical trials.